Hello, my name is Dr. Jia Ruan. I'm a lymphoma specialist at Weill Cornell Medicine and New York Presbyterian Hospital. My research interest is to design new treatment for rare subtypes of non-Hodgkin's lymphoma, including mantle cell lymphoma and T-cell lymphoma. I lead the clinical studies for T-cell lymphoma at Cornell. The future of T-cell lymphoma management looks promising and increasingly personalized, driven by precision medicine knowledge of lymphomagenesis and availability of a growing list of targeted agents that are active in T-cell lymphoma. So just a quick reminder that T-cell lymphomas are heterogeneous and uncommon, representing just 5 to 10%, which is a small fraction of non-Hodgkin's lymphoma, but has over 30 different subtypes based on cell of origin, driver mutations, and clinical presentation. The overall objective, therefore, for T-cell lymphoma management is to develop frontline regimen optimized for T-cell subtypes to improve CR and remission duration, as well as to design treatment for relapsed refractory disease that offer very high quality and durable disease control to preserve quality of life. So towards that end, biological agents available in clinical practice and in clinical experimentation are beginning to make a big difference in T-cell management landscape. For example, CD30 expression can be targeted by anti-CD30 antibody drug conjugate, brentuximab vedotin. The addition of BV to CHOP-based chemotherapy backbone has shown to improve survival in CD30 PTCL, in particular anaplastic large cell lymphoma, which has led to the practice-changing approval of incorporating BV in frontline treatment setting. Other important targeted agents with approval in T-cell lymphoma include epigenetic modifying agents such as HDAC inhibitors of romidepsin and belinostat, antifolate praloxate, and anti-CCR4 monoclonal antibody mogamulizumab. There are a number of novel targeted agents currently under clinical development that have shown promising activities. They include DNA demethylation agent azacitidine, histone demethylation EZH1/2 inhibitor vilometostat, PI3 kinase inhibitors such as duvelisib, JAK inhibitor such as serdulatinib. Preliminary data suggest that these agents offer solid response rate, in general over 50% as single agent and with tolerable side effect profile, as well as easy oral administration for chronic use. The frontiers of T-cell studies are to optimize novel agent combination for both initial and subsequent lines of treatment, guided by biomarker selection. For example, novel agent combinations such as romidepsin plus lenalidomide, romidepsin plus azacitidine are shown to be more effective than single agent alone, particularly for angioimmunoblastic T-cell lymphoma subtype derived from follicular helper T-cells, which have genetic mutations that make them susceptible for epigenetic intervention. We have studied the combination of oral azacitidine with CHOP, which showed excellent CR rate and tolerability. A US intergroup is currently underway to investigate whether addition of azacitidine or PI3 kinase inhibitor duvelisib to CHOP-based chemotherapy backbone would improve outcome compared with CHOP alone. So therefore, as you can see, there are a lot of work needs to be done, but I'm encouraged and hopeful for the future of T-cell lymphoma management. I hope to share additional knowledge and findings with you again soon. Thank you so much for your attention.